Good morning, everyone. This is Lynn from Biomarker Technologies. Also, we're known as BMKGENE. So first of all, welcome to today's webinar, and it's really great to see many of you joining us for this interactive session on advancing agri-food microbiome research with high-throughput sequencing. So I'm delighted to introduce our speaker today, Dr. Ana Villasanta, a field application specialist at BMKGENE, and she holds a PhD degree in bioengineering with deep expertise in synthetic biology, microbial surveillance, and extensive experience spanning both wet lab work and bioinformatics. So also, she has many years of experience in providing tailored solutions and technical troubleshooting around various applications of sequencing technologies. So over the next hour, we will walk through the latest sequencing strategies shaping agri-food and environmental microbiome research, from metabarcoding to multi-omics approaches, plus practical tips on picking the right workflow for your projects and with real case studies along the way. So we will also leave some time for a live Q&A session later, and please do keep your questions ready. So with that, I will hand over to Dr. Ana Villasanta. Welcome, Ana.
Hello, everyone. Hope you're having a nice morning. It's great to be here today. So first of all, I would like to thank the IRTA Institute and specifically Mari Hutner for organizing this webinar. So today we're going to talk about these high-throughput sequencing technologies and how you can use them to advance your agri-food microbiome research. So during this talk, the first section will be focused on these different technologies and talking a little bit about each one. Then we'll look at how a sequencing experiment, what is its regular workflow. And we'll also focus on a bioinformatics platform that we at BMKGENE have available so that you can analyze your results online. We'll also look at a featured publication of these omics strategies. And finally, we'll look at the BMKGENE's sequencing services portfolio. So when we want to do agri-food microbiome research, the strategy that was used for a long time and up until recently was culturomics. So the idea was just taking a swab of a sample of a river, a soil, a plant, etc., and swabbing it in media, and then trying to isolate the colonies in different selective and differential culture media to be able to identify the microbes there. But of course, as you can imagine, this is a very labor-intensive method, and it also has the major caveat that some microbes that exist in nature, they are not cultivable in the lab, so these were not identified using these methods. So nowadays we have all these different technologies that can be used to study the agri-food microbiomes that are much easier to implement, not so labor-intensive. And that's what we're going to talk about today. So the first one is amplicon sequencing, and what this tells us is who is there, which microbes are there. Then we have shotgun metagenomics, which not only tells us who is there, but also what they can do. Metatranscriptomics is focused on the RNA, and it tells us what the microbes are doing in that moment that we sampled. And metabolomics tells us which metabolites the community is producing. Ideally, we then have a multi-omics integration that enables, just like that, to integrate the different data sets to understand how the system is working. So now let's go a bit more into detail into each one of these. Starting with amplicon. This is also called metabarcoding, so you may be more familiar with this name. The idea is to sequence hypervariable regions of genetic markers. And usually this is 16S for bacteria, 18S for eukaryotes, and 18S for fungi. And the most classical approach, let's say, or the one that is still used a lot is, for example, looking at the 16S gene, since it's somewhat big, it's 1.5 kb. What people would do is they would amplify, for example, the V3, V4 region, and this region is between 500 to 600 base pairs. So this meant that since it's a small fragment, it can then be sequenced in Illumina, which is a sequencing technology for short reads. But nowadays it's becoming more common that instead of just focusing on the V3, V4, we would just amplify the entire 16S gene, and we can then sequence it in a long read technology such as PacBio. And of course, this can be done also for the full-length 18S, ITS. So you might wonder why do these shifts from short reads to long reads? So one major factor in here is the annotation rate. And this is a study from 2019 that performed an in silico experiment using 16S sequences in a database and constructing a taxonomy tree, either using the full-length 16S sequence or using just sections of it. And you can see this on the right side of the slide, that when we are looking at the full 16S gene, we can get a phylogenetic tree that has a very short amount of sequences that are unclassified at the species level. That means that looking at that entire 16S, we can say this is, let's say, an E. coli. We can say at the species level instead of just staying at the genus level. But if we're doing that, just looking at the V3, V5, so just looking at small regions of the 16S, we are not able to have such a good annotation rate. So we have lots of sequences in this case that we cannot know the species. We can know the genus, but not the species. So this means that PacBio enables a higher taxonomic annotation rate at the species level. And based on our in-house data, we know that this can be up to 12 times more at the species level when we compare PacBio to Illumina. But annotation rate is not the only thing. Another important factor is the ability to capture the taxonomic diversity of a sample. So in this study from last year, the researchers were aiming to compare different platforms to be used in 16S soil projects. So they compared PacBio, Nanopore, and Illumina. And you can see here, this is basically the different phyla that were identified in those soil samples. And you can see here when you look at PacBio 16S, the full-length 16S, and when you compare to Illumina V3, V4, that in fact there is a big difference. There are many phyla that were not identified with Illumina. And even when we compare PacBio to Nanopore, the diversity is somewhat similar, but what they found is when we're looking at low abundance taxa, PacBio was still the one that was able to identify those in a higher amount when compared to Nanopore. So I hope with this I could show you some convincing notes of how PacBio really is a good technology to be used for metabarcoding studies. But of course, since studies in metabarcoding using short regions of these marker genes and using Illumina are very well established, it's also important sometimes to continue using these protocols in order to be able to compare results obtained now to results that were obtained some years ago. So in this sense, at BMKGENE, we offer both options. We can do short read sequencing, so focusing only on those small sections of the 16S, 18S gene. And in this case, we use Illumina 6000, Namasec 6000, and we provide this different tags package. I would like to highlight what tags are here, what they mean. So basically a tag would be a read pair that passed all the quality control filters and it was successfully assembled into the target region. So when we say that you will get 50K tags, you will get 50K target regions that are completely passing these filters, which is higher, or let's say better, than 50K paired and reads. In the case of PacBio, we also offer a tag package. We don't need that many tags with PacBio because sequencing saturation is achieved with a lower tag number than with Illumina. And in both cases, an important thing to say here is if you send us the DNA and it passes the quality control in our lab, so if it's in good condition and we can amplify the target region with a good band, this means that we will guarantee you this contract tag number. And I think this is important because, well, you basically need a good amount of data in all your samples so that you can then have a robust analysis. We also provide bioinformatic analysis. This is really dependent on what each researcher prefers. Some researchers prefer their own in-house bioinformatics, really depends. So with amplicon sequencing, we can do mainly two kinds of analysis. We can do OTU, which basically we are clustering sequences that are highly similar. But there's also another kind of analysis, which is amplicon sequence variant, or ASV, that has become more common nowadays. And this uses a denoising model to handle the sequences. So both are available, again, depending on the preference of each researcher. And doing this analysis will give us the taxa that are present in each sample and also the relative abundance of those taxa. And that's what enables us to then perform all the downstream analysis that I'll show you here. So the first downstream analysis is just looking at the taxonomic annotation results and providing these different visualization plots and clustering plots so that you can just look at it in a more intuitive way, let's say. So this is a very common plot to see in metabarcoding projects, where you see here, these are the samples, and these are the relative abundance. It can be a phyla, for example. So the blue means one phyla, the orange is another phylum, etc., etc. So just by looking at this, it's already intuitive to see the different abundance of the different samples. And these plots can be seen at a different level, can be seen at the phylum, family, order, at the genus level, etc. Then we provide alpha diversity analysis. Here, alpha diversity can be used to look at the inner complexity of the samples. So you can see some samples are more complex than others, which means that they should have a more diverse microbiome, etc. Then we also do beta diversity analysis, and here the idea is to try to compare the samples or among themselves. So try to find the samples that are closest to each other than others in terms of complexity. And PCOA is a very common way to look at it. Then we have the differential analysis. This is usually I would say the main section, because this is where we can find the taxa or the microbes that are more abundant in groups of samples, one group of samples versus the other. And we do this with statistical testing, but also with tools for biomarker discovery, such as LEFS, which is the result I'm showing you here. Then we also carry out correlation analysis. And correlation analysis, like this graph here, can tell us if we have some microbes in your sample that are correlated among themselves. So if when one appears in a high amount, usually the other one also appears in a high amount, and this would be a positive correlation. A negative correlation is the other way around. In this section, there is also a part which is environmental correlation analysis, which is usually interesting for researchers that are doing environmental studies, because it might be interesting to try to correlate environmental variables such as pH, humidity, amount of heavy metals, organic load, etc., with the microbes that are present in your samples. And then we have a section that is based on functional gene prediction. So what we do here is we already know the taxonomic distribution of the sample. So we know how much of each bacteria or fungi is there. And then using a database, we can infer the function abundance in those samples. So we can have something like this, where we can say, okay, but in group A, there's a higher abundance of carbohydrate metabolism functionality than in group B. But of course, I think it's worth noting that this is a prediction. It's not based on actually sequencing those functional genes. So if someone is interested in this, or if the result is something that it's interesting, it opens the avenues for new projects, I would say that the next step is go to metagenomics. And in metagenomics, in fact, we can sequence the functionality. So we not only know who is there, but also what they can do. So with metagenomics, the sequencing workflow is simpler. The idea here is we just take the sample, we extract the DNA, and then we prepare the library from it. And as I mentioned previously, using those DNA reads, so those sequences, we are able to know the microbial community composition, so we can know which microbes are there, but we also know the genetic functions because we can profile the functional genes that are present there in different databases and assess the functionality. This also means that we can then focus on more specific things. For example, we can look at the metabolic network, or we can also look at signature or marker genes. And this includes looking at antibiotic resistance, heavy metal resistance, these kinds of marker genes that are often relevant in these studies. In some circumstances, it may also be possible or of interest to be able to isolate single bacterial genomes from this complex mixture that has bits of multiple genomes of bacteria, fungi, and everything else that is present there. Regarding to the sequencing technologies we are using, we are using these 4 technologies, so we can use Illumina, DNBSEQ, Nanopore, and PacBio Heavy. So now I would like to talk a little bit on why use each one of these. So just as we've talked previously, of course, it's important to understand why go to long-read sequencing instead of using short-read, which is always cheaper. And this is particularly relevant when we're talking about wanting to assemble single genomes from that complex mixture. So in these cases, this is, I would say, maybe a difficult task bioinformatically, because it's something like this where we have a bunch of Legos with different colors and shapes, and we're trying to find the ones with the same color and trying to put them together to form a structure. So when we are doing just short reads, this means we are getting reads that have 150 base pairs, and then we assemble into contigs. Then we take the contigs and we bin them, meaning we put contigs that are coming from the same microbe in one bin, and then the other microbe in another bin, etc., etc. And this is what's called binning. And then doing this, we can obtain a genome that will, of course, have gaps because we have one part of the genome, then the other, but it will have gaps. If we also use long reads, this means that the contigs that we are able to assemble are longer, and it also means we will have less contigs that are longer. So it's much easier to then do the binning and improve it, and to get genomes that have less gaps. That is the general idea. But besides the general idea, of course, it's always important to then quantify this and put this into numbers and actually validate and benchmark the different technologies. So this study in 22 did this. It benchmarked these 4 technologies to do microbial metagenomics. There are 3 main parameters that were assessed here that are relevant for metagenomic studies. One is the continuity, so it's the presence of gaps, as I just mentioned. And of course, with short reads, the sample genomes are always, they always have more gaps. Then with long reads, PacBio is the technology that obtains the most continuous assembly, and that is followed by Nanopore. Another parameter that is important here is the genome recovery. For this, I have to explain that in this study, the authors were using a mock community, so that meant they know the microbes that were present in the community. So after sequencing and assembling, they then looked at which microbes were identified with each technology, and also the percentage of the genome that was identified. So that gave a percentage in the terms of genome recovery. And short reads got somewhat a good recovery. I think it was around 60%. Then Nanopore got 66, something like this, and PacBio got the highest recovery. And the last parameter is accuracy, and that is basically taking the reads and aligning them against the reference genomes of the microbes that we know are there, and seeing if we have a lot of errors in our reads. So in this is where short reads are really nice because they have a good accuracy. So the error rate was low, and PacBio also has a good accuracy. So the error rate was also low. Nanopore is the middle one here because although it has long reads, so the assemblies are nice and more continuous, but the accuracy is lower. So based on these characteristics of each technology, we can then find which are the better options depending on the research purpose. So I think we can maybe in general, divide that metagenomic studies can be focused on just doing a general profiling of the taxonomy, so general profiling of the microbes that are present there, and also the functions. And for this, we need contigs, but we don't need them to be very continuous genomes. We just need the contigs. So for these options, and if budget is a constraint, we can then use Illumina or the NBSEC, so the short reads, and this will be a good option. And in both cases, among these cases, the NBSEC is the lowest cost option. If the purpose is doing the assembly of the genomes among that complex mixture, so this is called the metagenome assembled genomes, then it's best to include long reads in this. And here, using PacBio is the one that provides, as you can see, best continuity, recovery, and accuracy. But since it's also a bit more expensive, if budget is a constraint, we have the option of doing Nanopore. So as you can see, Nanopore has an accuracy problem, not say a problem, but it's not as good. So the strategy that we use is we assemble with Nanopore to get longer contigs, and then we use NGS reads from Illumina to correct the assembled genomes. So we can improve this accuracy, basically joining the best of both technologies. I would also just like to add that even if the idea of the project is to do general screening of the taxonomy and the functionality, although short reads are good for this, if you have the possibility of doing long reads, since the context will be longer, of course, the screening will also have a better quality. So in terms of the service we provide at BMKGENE, we generally recommend to obtain a minimum of 6 gigas, up to 20, and sometimes even 40, depending on the complexity of the sample. And if you are interested in looking at those low abundance microbes, if yes, then the data volume needs to be high. And the bioinformatic analysis is very similar to what I've shown previously for amplicon sequencing. So we have the beta diversity, the differential analysis, the correlation analysis, it's very similar. The main difference is with amplicon, we only look at taxonomy, and with metagenomics, we have a section for taxonomy and another section for functional diversity. So now going into metatranscriptomics, where we are looking at what the microbes are doing, and this is using RNA. So metatranscriptomics captures the complete pool of RNA of a sample, and this reflects the actively expressed microbial functions. So we just take a sample, we extract the RNA, and then we prepare the library for sequencing. We need to apply a depletion method to remove the ribosomal RNA from the sample. This is because ribosomal RNA can be even higher than 90% of a sample, and it's not interesting to sequence it. So we want to remove it so that we can then just sequencing everything else. The ribodepletion that we apply removes ribosomal RNA from bacteria and also for eukaryotes. The recommended data volume is usually 12 to 20 giga. For viral studies, it's usually higher than 20 giga because viruses are present in low amounts, so the read percentage can be even lower than 1% depending on the sample and the pre-treatment that is done. This is the bioinformatic package that we offer. Also in continuity to what we have been talking, it is similar to metagenomics. We look at function, we look at taxonomy. We have all these different downstream analyses of function and taxonomy. But then, since we're talking about RNA, we can also look at gene expression and find the differentially expressed genes. So we get volcano plots like this. when we can look at the up and down regulated genes and then do some functional enrichment of these genes with gene ontology and KEGG. The final technology here is metabolomics, so looking at what the community is producing. This is done using non-target metabolomics, which uses high-resolution mass spectrometry to perform an unbiased screening of metabolites. And here, the key is unbiased, so we don't have any information on the metabolites that we want to quantify and to identify. So the workflow here is extracting the metabolites from the sample, injecting into LC-MS. Then we get the peaks. And with the peaks of MSMS, we can identify the substances by comparing them to public and in-house databases. And we can also calculate the relative abundance of the different substances. Then our bioinformatic analysis has these kinds of volcano plots, so looking at the differentially abundant metabolites, and also a functional annotation, so finding which pathways are more enriched in A versus B. And finally, the ideal would be able to integrate these different data sets to know how the system works. So at BMKGENE, we have two multi-omics packages that are relevant for this case, which are Amplicon plus metabolomics and metagenomics plus metabolomics. And the analysis that we do here is basically data integration methods that integrate these two data sets. So it has a global similarity analysis to see if the Amplicon and the metabolomics data sets are similar. We also do clustering to find biological associations, and this heat map here is an example where you have the microbial taxa here, and then here you have the metabolites, and you can see the two clusters. We also do multivariate structure analysis to find correlations among the data sets, and biomarker discovery to find the biomarkers in each groups in clusters. In the case of doing metagenomics plus metabolomics, we are also able to do some KEGG pathway analysis of the differential abundant metabolites and functional genes. So you can see here the two data sets, metabolomics and metagenomics, the different pathways, and if they are similar or not, the fold change, etc. So now moving on to a more practical aspect of this. So how does a sequencing experiment work? This is the typical workflow when you work with us. You can send us a sample to extract DNA or RNA, or you can already send us the DNA or RNA. We will aid you with the shipment and explain how to package the materials and etc. And then once we receive the samples for extraction or already extracted, we perform the sample quality control. And this will depend on the service, but in general, for DNA, we want to look at the quality, the quantity, the purity, etc. With amplicon sequencing, we want to amplify the target region and then see if it was amplified by capillary electrophoresis or by gel electrophoresis. With RNA, we want to look at the concentration, the purity, and the integrity, because integrity with RNA projects is very important to be able to have success in it. Once we have done this, we send the researcher the quality control report, and the researcher can then decide if they want to proceed with the sequencing project, so with the library preparation. If in some cases the sample is not, let's say, perfect, it doesn't have the perfect concentration or amount, we can then provide you some advice on what to do if you can decide to proceed with some risk, or if it's best to really prepare new samples to send, if there's something else we can do to try to handle this. Then we prepare the library and we sequence it. After obtaining the data, we perform quality control just to look at the data volume, the quality of the reads, etc. And we provide you with the data quality control report. After that, we can carry out the bioinformatic analysis, and this depends on what each researcher prefers. And going into the bioinformatics, This is where I would like to highlight the platform that we have online. This platform enables you to have the sequencing results there and then carry out customized analysis. So for researchers that decide to do amplicon and metagenomics with us and doing the bioinformatic analysis with us, we give free access to this platform where we put there the report with the results. And the researcher can then carry out a number of tasks in this platform. The important thing here is it doesn't require any coding experience or bioinformatics expertise. As you can see, this has a user interface, a graphical interface, so no coding. It's through a web browser, so you don't need a special program or anything, just going to a web page and logging in. You can generate publication-ready figures in image like this one. And I think this is the most relevant one from the scientific point of view, is that you can customize your analysis without rerunning the full pipeline. So just giving an example here, if you want to do alpha diversity, you can then do different alpha diversity methods in the BMKCloud platform. There are also some other examples. You might want to change the sample selections, or you might want to do different grouping schemes of your samples. In some cases, the metabarcoding projects, you might have a very abundant taxa in your sample. So let's say there's a species there that is completely dominating the data set and you are not able to do proper analysis. So you just remove it so that you can then carry out the analysis, since you already know that that species will be there. You can also do advanced visualization and change the images, aesthetics, etc., etc. So now I would like to show a featured publication. So this was a publication that came out, that was published from a researcher that used our amplicon sequencing services. So this project was focused on aerobic digestion of wastewater treatment plant sludge. And this can sometimes have a suboptimal methane in biogas production. So what the researchers wanted to see was that if they can improve it by digesting the sludge with these factory residues, and the idea here is that it would provide additional nutrients and stimulate the microbial activity. But also they wanted to see if adding a current sensing molecule would further enhance the stability and the degradation of the complex substrates. So in order to study the mechanisms behind this, the researchers performed amplicon sequencing of full-length 16S and ITS with the Pacbiome. So there were three main questions here, research questions. The first was doing the digestion with cheese waste and adding the current sensing, if it does improve the methane and biogas production, and the researchers saw that yes. And then they wanted to see when the current sensing molecule should be added to maximize the performance. And they found that at 4 days of cultivation, it was, when they got peak performance, so peak production of methane and biogas. So the next part is where amplicon sequencing entered, because now the goal was to see what is the current sensing molecule doing here and how the community is shifting when they add the current sensing molecule. So one thing they saw is that there was an enrichment in biofilm-associated families. You can find here, this is samples that were collected at the end of the cultivation. This is the control, and this is when the current sensing molecule was added at day 0, 4, and 8. And you can, looking at this comparison, that you have a higher abundance of families that are biofilm associated when you add the current sensing, which is expected because the current sensing increases the cooperation among bacteria. Then the researchers were also able to find that there was an increased abundance of syntrophic taxa, so also involved in bacterial cooperation. And these taxa are mainly involved in these metabolic activities and interspecies metabolite exchange. So this may be behind the improvement in biogas and methane production. particularly when the current sensing molecule is added at day 4, so the optimal condition is when they saw an increase in Dermatoga, which is specific bacterial genus that may be of interest since it may be behind this improved production. Using this functional prediction analysis, it was possible to then identify some functional categories that are increased in abundance when the coronal sensing molecule is added. And also unexpectedly, when the coronal sensing molecule is added, it was possible to identify fungi, while not adding it meant that no fungi were identified, which is somewhat surprising, but also interesting. So this is just one example of a published paper that came out of services that we provide. But just in the last year, we had over 400 publications using our microbiome that came out of our microbiomic services. In 2004 and 24, there were over 250 publications that use our microbiomic services. And I just put here a table with a few examples from this year, but it's not complete. It's just a few examples. So finally, just to say that we also, in microbial genomics, we also are able to provide other services, and this includes assembling de novo microbial genomes of bacteria and fungi. And we can also carry out resequencing projects, so whole genome sequencing of bacteria and fungi. And besides our microbial genomics, we also have other sequencing sections that we can provide. So in transcriptomics and epigenetics, we can do RNA-seq, ATAC-seq, Hi-C, long non-coding RNA-seq, etc. In plant and animal genomics, we can do assembly of reference genomes, and this is where we also have lots of expertise, namely with plants which have big genomes, and sometimes it's a bit a difficult task, and we have lots of experience with this and a very expert bioinformatics team to handle these challenges. And we can also do population genomic studies. We also can do human genetics and single-cell omics. This includes single-cell sequencing, but also spatial transcriptomics, where we have our in-house developed platform, BMKMano, to do spatial transcriptomics. Then we have BMKCloud, as I just mentioned. So this is our bioinformatics platform online. In order to carry out all these different services, it means we also need the different technologies in our labs. So we have Illumina, PacBio, Nanopore, and DNP-seq available. We also have 10x Genomics to do single-cell sequencing. And we have a bioinformatics team that can handle all of this. So that is all that I have to talk today. Thank you very much for listening. And now we are open to questions.
Thank you so much, Anna, for the comprehensive presentation. And now we will open the door to a live Q&A session. So please feel free to type your questions in the Q&A window. And actually, I already see one of them here. So saying nice presentation. The question is about the sample requirements. So what should be the minimum DNA or RNA concentration for each of the 4 technologies to pass quality control?
Okay. So this depends on the different technologies, of course. So I can give you some numbers. For amplicon sequencing, the concentration we require is not very high. It's just 1 or 5 nanograms, depending if it's Illumina or PacBio. And the amount is 200 or 300 nanograms for Illumina or PacBio. But it is important that for amplicon sequencing, what matters most is not so much the amount of DNA that you have, but if we can amplify it. So if you say that you want to do 16S sequencing, we are doing the primers to amplify it. And then we do capillary electrophoresis or gel electrophoresis to see if we have a band. And that's what matters, because if we have a band, we can continue with the project. If not, it's not possible. For metagenomics, we have a very low requirement with the Illumina, only 30 nanograms. And with metatranscriptomics, this requirement is higher, but I think this is always the case when we're talking about RNA. So RNA projects require a lot and also require a good integrity. So it should be 1 microgram, and the RNA integrity number should be higher than 4.5 to be able to do this.
Okay, thank you for the answer. And another question is asking about metabarcoding. So, do you only do metabarcoding with 16S V3 plus V4 region? Uh, is that I work with plant microbiome communities, but knowing the most common 16S primer pairs can also cause some plant amplification as well. So, do you have any comments on that?
Yeah, so we have a full list of primers that are available. I focus on the 16SB3 before because it's the most common, but we have a very big list of primers that we can amplify. Really dependent on each in each project preference. In the case of endophytic communities, we have some primer pairs specific for that. For 16S, we have for the V5, V7, which avoids co-amplification of plants.
Okay, good. Thank you for the detailed instruction. And we may only have two more questions because of the time. So I got another question about the database. So which database do you use to analyze shotgun metagenomic results? Thanks a lot.
Okay. Yeah, so that's a very important question because it really will affect the analysis. So for the taxonomy part, we do it with NCBI. We also do, and so then for the functional part, we also do with NCBI, with KEG, with the, I think, FAM. So these are the general databases for functionality. Then we also use specific databases. That is those marker genes that I was mentioning. So we use CARD for antibiotic resistance. We use also for heavy metals databases. We also use for carbohydrate. So we have this distinction between general functional databases and more specific marker genes databases. So in the end, you get a functional analysis that has all of these different results.
Okay, thank you. So we only have time for the last one. The question is about analysis as well. So how do you calculate beta diversity? And can I use the platform to these calculations? I think they mentioned about the BMKCloud platform. Yeah.
Okay, so starting with the last part, yes, for sure you can use BMKCloud to do the analysis. In our standard bioinformatic package, we provide beta diversity analysis using 4 indexes, but then in BMKCloud you can also do some more customized and regenerate the graphs, etc. The 4 indexes that we use are Bray-Curtis, Jacquard, and then Unifrac. And the main distinctions here is that it can be weighted or unweighted. So we can just consider if the species is present or not, or we can also consider if it's relative abundance. We can then also just consider the presence or absence of the species, or also the phylogenetic relationships. So, because this will contribute to the complexity of the sample if the species are somewhat close together in terms of phylogeny.
Thank you. And by the end, we also received a thank you note from Maria. It's really nice. Maria Hutner saying, "Hi, this is Maria Hutner, and I just want to thank Anna and Jose for the presentation and explanation and for organizing the webinar. Thank you. And thank you to Maria for making this happen. And again, I would like to thank Anna for the very detailed answers and also the pleasant presentation. It's been a really pleasure interacting with this. So for all the attendees today, if you have any follow-up inquiries or questions about sequencing strategy or data analysis platform, anything, please do not hesitate to reach out to our team directly. And we're here to support your research every step of the way. So I guess that concludes our session today. Thank you so much for joining us, and we wish you all have a very best in your research and also have a wonderful day. Thank you.